Good afternoon, everyone. I am Dr. Mohit Chaudhary. This is TransConnect, and we are in July 2024. And today we'll be discussing about a very important topic called as transfusing in a platelet refractory patient. So I'm going to talk about the introduction of the same, what platelet refractoriness is, since HLA and HPA, that is the human platelet antigen and the human leukocyte antigen are involved in it. What are the basics of them? What are the tools available for diagnosing? What is a platelet crossmatch? And how do we approach in a case of platelet refractoriness along with our experience, how we have dealt with it? So platelets are critical mediator of hemostasis. They have a variety of immunogenic proteins which are unique to the platelet themselves or shared with other cell types. Now, platelet refractoriness, what it is a major barrier to efficacious platelet transfusion, and it is associated with bleeding, longer hospital stays, higher input costs, and decreased overall survival. So it's very important that we diagnose it and we are able to provide the platelets in these platelet refractory patients. However, platelet alloimmunization refers to the de novo development of alloantibodies which are capable of binding the platelet antigens. So you may have alloimmunization, you may have platelet refractoriness. These two terms are not interchangeably used. They are different terms. You need to know about them. So you can have a platelet refractoriness but not platelet alloimmunization, and you may similarly have platelet alloimmunization but not platelet refractoriness. So when you have alloimmunization, what are the causes of alloimmunization? It's basically the antigen exposure associated with leukocyte in three cases, that is a transfusion, pregnancy, or transplantation, which resolves spontaneously if there is no further alloimmunization. Or what it can does is it can cause two problems. One is the humoral transplant rejection, that is the antibody mediated rejection, or you can also have refractoriness to the platelets. So these antibodies can react with class 1 HLA, ABH antigen, or the platelet antigen that exists on the surface of the platelets. So these two terms are different, platelet alloimmunization and platelet refractoriness. Alloimmunization basically means it involves the production of the antibodies against the donor platelets. However, refractoriness is a clinical condition in which patients do not achieve the anticipated platelet count increment following platelet transfusion. Now, this platelet refractoriness can be because of immune causes or non-immune causes. When it is immune causes, it's basically because of the antibodies that are present against whether the HLA, HPA, or ABH antigens. So basically, you can have a platelet alloimmunization and not have platelet refractoriness, and vice versa. So now what are these platelets? These platelets express, we know what are platelets, but what do they express? They express a number of antigens that are relevant to the alloimmunization and refractoriness, and these include HLA and HPA antigens. Now you can see that here in this diagram, this is the HLA antigen, and generally it is the platelet express HLA class 1, that is HLA A and B mostly, not the HLA C. And you have the HPA antigens, which are on various glycoproteins which are there. So you can see there is glycoprotein 1A, there is glycoprotein 2B3A, you have glycoprotein 1B9. So all of these glycoproteins have various HPA antigens, that is the human platelet antigens which are present, which may be HPA 5, 18, 13, based on the wherever they are found and in what order they are found. So these are various antigens that are there on the glycoproteins, that is the platelet glycoproteins. So coming to specifically what HLA is, just a brief outline about what HLA is. Of course, we have a different uh, episode itself on human leukocytes and basics, human leukocyte antigen and basics of HLA. So what is an HLA? HLA is a misnomer. Why do I call it as a misnomer? Because it is not just present on human, it is not just present in leukocyte. In fact, class 1 is present on all nucleated cells and platelets, and class 2 is present on the antigen presenting cells. So you are not, they are not restricted to leukocyte. In fact, most of the cells of the body, nucleated cells, if you see, are having HLA class 1. So it is a misnomer. So, and also you must have come across a term called MHC, that is the major histocompatibility complex. So HLA and MHC are used interchangeably, and you can call either or, but HLA is a more commoner term. So it is critical for immunological specificity, histocompatibility, and susceptibility for autoimmune diseases. Basically, it is a region, it's a genetic region that is located on chromosome 6, small arm of chromosome 6, and it has got around 140 genes, 20% of which have functions in the immunity. So HLA class 1 antigen, if you see, I have already spoken about it. It is present on the all nucleated cells and platelets. So HLA class 1 antigens are present on all nucleated cells and the platelets. And in this case, HLA class 1 is what we are dealing with. So HLA class 1, we have the HLA class A, B, and C. In there, A and B are more important. HLA-C are relatively less polymorphic. So similarly, you have the human platelet antigens that are also expressed on the platelets. These are basically the glycoprotein found on the surface of the platelets. Now they have a biallelic system, and around 24 platelet specific alloantigens have been described. And since you have an antigen, you have an antibody against this antigen, and these are called as anti-HPA antibodies. So these are various glycoprotein, and these are the antigens on these glycoproteins. So this human platelet and alloantigens, this is a list of the human platelet and alloantigens and glycoprotein which have been there for right now and discovered. HPA1A, if you see, is the most common. Now it is a biallelic system, so basically you have 1A and 1B. 1A is the more commoner allele, and the B is the not so common allele. So 1A, if you see amongst all the HPA, 1A is the most common. Its synonyms include ZWA, PLA1, located on glycoprotein 3A platelet glycoprotein, and it has been also implicated in 80% of neonatal alloimmune thrombocytopenia. So HPA1A is the most commonest of all, and it has been now discovered that this is not biallelic. This one antigen has 3 alleles, and it is called a triallelic, in fact. Other antigens that are more important are HPA5A, which is second most common in Caucasian, HPA4, which is relatively more common in Asians. So 97% of the adult phenotype is HPA1A, that is positive for 1A. 69% are homozygous for 1A, and 28% are heterozygous at least. 3% of adults are homozygous for HPA1BA. So if HPA1BA person receives the blood from a person who is HPA1A positive, that is most of the donors who will be having one antigen of HPA1A at least. So if you receive blood from an HPA1A positive uh, donor, obviously you are going to make antibodies against HPA1A, and anti-HPA1A antibodies will be formed. So this is the basics of the human platelet alloantigens and antibodies. So now, when do you need platelet transfusion? So there are two indications, whether it's prophylactic or therapeutic. So prophylactic is when you prevent the bleeding complication due to low platelet count, and therapeutic is when there is an active bleeding or low counts and you need to intervene and act therapeutically. So it is said that preventive maintenance of vascular integrity requires at least 7,100 platelets per microliter per day, and this is constant irrespective of the platelet count. So what is platelet refractoriness? Platelet refractoriness is defined as repeated suboptimal response to platelet transfusion with lower than expected post-transfusion count increments. So this means that literally that whenever you give a platelet, that is whether it's a random donor platelet or a single donor platelet, you expect a bump or an increase in the platelet count. Now, if that response that you're getting is suboptimal, it may be platelet refractoriness. Now, to measure this, there are few calculations that can be made. There is one of them is a corrected count increment, which I'll be talking about in brief. So CCI or corrected count increment less than 7,500 at 10 minutes to 1 hour post transfusion and less than 5,018 to 24 hours after two sequential ABO compatible platelet transfusion points towards platelet refractoriness. Now, there is a difference here. There are other measurements which we'll be talking and discussing later. But just to reiterate that basically platelet refractoriness, it's not just that you keep doing these counts and wait for the sequential ABO compatible platelet transfusion and then you actually go and measure. If you're a clinician, you would see that you can anticipate that there is going to be platelet refractoriness in these patients because the clinical signs and symptoms point towards there is platelet count which is low, and you don't need to wait for that. You start going for the investigations and you basically try to start investigating. Otherwise, you will lose a very, very critical time in that. And that is why history and clinical experience is utmost important, especially in patients which are more prone to platelet refractoriness. So, of course, you have to make those tests, you have to send the labs, and that will actually point towards the platelet refractoriness. But your clinical judgment is imperative here. Now, there are various causes for platelet refractoriness. The labs are going to detect the immune causes or you are going to detect those antibodies. But please remember, most common cause is not immunological cause. It's in fact non-immunological cause. 60 to 80% of the cases are non-immune causes. And what are those? It can be because of fever, it can be infection, some medication, DIC, splenomegaly. So all of these point towards non-immune causes, whereas immune causes is what we are concerned with, and we have to look for the platelet antibodies. They can be HLA antibodies, HPA antibodies, and ABO antibodies. So these all can cause platelet refractoriness. Now, the calculation that I was referring to, there are two calculations that you can do. One is the corrected count increment and other is the percentage platelet recovery. But how do you do that? You have to calculate the platelet increment that first. So what is that? It is the basically you subtract the post-transfusion platelet count from the and pre-transfusion platelet count. So P2 minus P1 will give you a platelet increment, and then you calculate the CCI or corrected count increment. So that is basically the platelet increment into body surface area upon N into 100, where the body surface area in meters square and N is the number of platelet transfused. So if you have that platelet count available, the exact platelet count is unavailable, you can give it as 3 into 10 raised to power 11. So provided you are giving the required dose of platelets. So an acceptable response for CCI is greater than 7500 at 1 hour and after transfusion, or greater than 5000 at 20 to 24 hours after transfusion. Similarly, you can calculate the PPR, that is the percentage platelet recovery, which is platelet increment. Instead of body surface area, it will be total blood volume upon N into 100. So these are the three parameters that you need to know, or will point out whether you are actually dealing with platelet refractoriness or not. One of the things that we tend to miss is the platelet count itself. You have to give an adequate dose. So if a suboptimal dose is given, that is the standard dose of platelet or adult therapeutic dose of the platelet, which is 6 units of pooled whole blood derived platelet concentrate or 1 unit of apheresis platelet count, should give a bump or increase in the platelet increment of around 30,000 to 40,000 platelets per microliter 1 hour after platelet transfusion. So if you are not giving the adequate dose, you will not expect a increased count, and that you may think that it is platelet refractoriness, but actually the dosage or the platelet count has not risen because of the inadequate dose of platelets that has been transfused. Nonetheless, if that is okay, then you have to have two calculations. One is of them is corrected count increment and PPR. More often we use the corrected count increment, and this is basically the cutoff is less than 7500. So it is measured generally after 10 minutes to 1 hour after the completion of platelet unit, and lower values observed after 1 hour are indicated of immune mediated clearance, and after 24 hours, it is generally presumed to be because of the non-immune mediated platelet clearance. Similarly, you can calculate the PPR, platelet percentage recovery, which is similar to CCI, only it requires a patient blood volume to be estimated. And you can see that studies show average 1 hour PPR of approximately 66%, recovery less than 20 to 30% at 1 hour post transfusion indicates a refractory response. Now, what are the tools for approaching a case of platelet refractoriness? So there are various tools available. If you are a lab that does Luminex based testing, or suppose you are doing ELISA based testing based upon what are modalities that are available with you, you can do an HLA antibody screening test, or you can do these three modalities that are available. You can give an HLA HPA match platelets, but you require an HLA typing for that. Second is the antibody specificity prediction platelets, and third is through the platelet crossmatch. Of course, there is a newer modality that is PAC-LX, which I'll be discussing today, which is a Luminex-based test, which also tells you the specificity of the platelet antibodies, whether it is HLA or HPA. So how do you allocate the platelets? You have three modalities in the lab that are available. You can give an HLA match platelets, an antibody specificity prediction or ASP, or platelet cross-matching, like I said. So what are HLA match platelets? Basically, what you do here is you type both the donor and the patient for HLA or HPA, depending upon what you are dealing with, and if you have the facility. The problem with this is, so HLA itself is a costly affair. So if you're dealing with HLA class A and HLA class B and two alleles for each, so you are actually going to do for patient and donor, it's going to be 4 HLA typing that you're going to do. So it is an expensive thing, and turnaround time is okay, still tackled, but it requires resources and it is expensive. And this matching, if you're arbitrarily going to test for it, HLA is considered to be hyperpolymorphic. So if you're going to start with the matching, it requires large number of HLA type donor to actually have and match at both HLA and HLA-B. And there may be some rare HLA types or haplotype at times. So it is said that a registry of around 18,000 to 25,000 HLA type people is needed to actually have one match. So this requires recruitment of specific donor, and these also can be recruited, and then after that you have to do an apheresis procedure, which is in itself sometimes a problem. So large blood suppliers periodically have difficulty in identifying the HLA match, and there are different match grades like A, BIU, BIX, BTU, CD. These are all grades of matching, and basically because there is a cross-reacting antibody sometimes. So you need to know where the match is. But the problem here is the more HLA typing you do, the HLA class one you have to do the HLA class A, HLA-A and HLA-B. So it is relatively less expensive and sometimes futile because of the hyperpolymorphic nature of the HLA. Now, the second way you can do is antibody specificity prediction. That means you need to perform a test called a single antigen bead assay to identify the antibodies in the patient, and you need to know the antigen in the donor. So once you have the antibody specificity of the patient, then you need to just type the donor platelets and look virtually if the antigen and antibodies are present in the donor and patient. So that gives you indication whether the platelet lacking the antigen to which the antibodies are present can be provided or not provided. So this requires a registry of donor platelet antigens. If you have a registry, it becomes relatively much easier and does not require repeat testing of the patients. Only the donor can be tested. So because you have the donor uh, testing that is available, that is the donor typing, which will tell you about the HLA antigens. You just have the antibody status of the patient and you just need to compare it virtually. So basically this helps in moderately to highly sensitized patient, and the caveat in this is single antigen bead assay. First, you have to have resources. Secondly, you need to also take into account that these antibody have waxing and waning, and sometimes patient may have a blood transfusion in between. So in those cases, there may be some new antibodies that may be formed, and then you need to repeat the patient's antibody status and then compare with the HLA antigens in the donor. The third and the easiest way to start platelet immunobiology is platelet crossmatch. It is a simple crossmatch where you have a donor cell and patient serum, and there are various techniques available. Nowadays, most of us do it with the solid phase technology. Solid phase capture method is used to screen patients' plasma for platelet antibodies directing against HLA or other antigens on the platelet. So basically, a given plasma is tested against patient sample. Patient's platelet samples, donor platelets lacking reactivity in the assay are considered to be cross-match compatible. So basically you have the compatible units which can be issued and incompatible units, maybe few or all units depending upon how immunized the patient is. So advantages of platelet cross-match are it is easy, it is least expensive. You do not require, if you have a capture uh, process in place, you can do a platelet cross match. It is very fast. By the time you even start, preparing yourself for the HLA typing and HLA match platelets, you will see that by the time you do that, your platelet cross match is ready and you are ready with the compatible units. The other advantage is it detects both HLA and HPA antibodies. So this may be the only way to find compatible platelet in patients with antibodies other than against the HPA1A. So sometimes you require platelets because platelets is many a times an emergency, and that is where platelet cross match comes very handy. You don't have to go through a different process of HLA and HPA because it detects both. So a startup test in the platelet antibody testing, I would say that platelet cross match is the test. Fast, easy, less expensive. Only problem with platelet cross match is that sometimes when you're very immunized, then you are not able to provide the compatible units in platelet cross match. And it's a dictum that if you are immunized, more than 65% of them are, suppose your PR, your panel reactive antibodies test is greater than 65, then it's difficult to have a platelet cross match compatible. So in those cases, you have to resort to other modalities, whether it's a antibody specific city, or you can also go for an HLA HPA matched platelets. So disadvantage, like I said, it requires patient sample every time. It is difficult to support the patient with initial cross reactivity. Sometimes all units may come incompatible and it is not suitable for highly sensitized individuals. So what is the principle? It's a capture technology. So basically you have the donor platelets that are bound to the surface of microplate wells. You have the capture antibodies from patient or donor serum, and serum is incubated with the platelets that are coated onto the wells. So basically there is antigen coated well and you add the serum or plasma, you incubate them, wash them, and you add the indicator red cells. After that you centrifuge, and centrifuge what it does is it brings the indicator cell in contact with the antibodies bound to the platelets. So in a positive result, obviously the migration of the red cell to the bottom of the well is impeded because the anti-IgG bridges are formed between the indicator cells which are there, which we had added here, indicator cells and the platelet bound antibodies. So there is less migration, or there is impedance in the migration, and that is why a confluent layer may be formed. A confluent monolayer means strong positivity, and negativity actually means when there is a pellet that is formed. So this is a basic, very simple test, and which will give you detection of both HLA and HPA antibodies. So what are the advantages between the three methods that are there? HLA matched, of course, advantages. It is useful for very highly sensitized patients. But the problem is that you require a registry of around 18,000 to 25,000 donors. The restricted donor pool is there. HLA typing itself is expensive and does not help when platelet-specific alloantibodies are responsible for refractoriness. ASP, what it does is it widens the donor pool, does not require recipient HLA typing, but problem is does not help when platelet-specific alloantibodies are present for refractoriness. So ASP basically widens the pool, and it requires repeat antibody specificity testing to monitor the HLA and alloantibody profile and ensures continued provision of HLA compatible platelets. So moderate to highly sensitized individuals, you can have a ASP. Whereas platelet crossmatch, if you see, it has got a very rapid turnaround time. It's very fast, is not so expensive, detects both HLA and HPA antibodies. The only problem is when the individuals are highly sensitized, it does not work because all of the cross matches may come incompatible. So which tool is better? I would say none of them is better, or it depends upon how the person is, how your lab is, whether you have the resources. Initial startup may be a platelet, cross match, which will detect HLA and HPA, or you can even have a if you have the Luminex based platform in your laboratory, or have the facility of the same, you can go for a HLA antibody testing, that is a single antigen bead assay, and look for donor typing. Or you can go for, if you are a registry, you can go for a donor typing or HLA or HPA matched platelets. So depending upon what logistics, you can have that modality available for you. So now, how do you approach for platelet refractoriness? But before you do that, you need to know that the adequacy of the dose is very important. The dose of the platelets that you're giving, it should be an adult or depending on pediatric therapeutic dose, and that will give you an expected increment of 30,000 to 50,000 depending upon what you're using. Always rule out the non-immune causes like sepsis, DIC, splenomegaly. You should also look for quality of the platelets, whether the platelet quality is in place, the dose of the platelet, the age of the platelets. You can't have an expired platelets and expect that, okay, this is going to give you a good increment. So check the dosing, check for ABO mismatch, and then you would go for, if you anticipate platelet refractoriness and based on your clinical judgment, perform the CCI and PPR. So once that is established that, okay, this is the platelet refractoriness, you go for the calculated CCI, and if it is less than 7,500, you or greater than 7,500, this is the diagnostic algorithm you can follow. This is from Rossi. So you can go for a platelet specific antibody test, HLA antibody screen, and cross-match platelets. So basically, HLA antibody screen, what it does is it is going to detect whether there is some HLA antibody present or not present. So if only HLA antibody test is positive, then you go for an HLA antibody identification and order, if not available, HLA class 1 typing by low resolution. So you look for antibody and see whether you can give HLA matched or HLA negative platelets. Similarly, if HLA antibody screen is negative, but your platelet specific antibody testing is positive, you look for XPA typing, you look for, or you do go for a cross match platelets, which will detect both HLA and HPA. The third scenario is when HLA antibody and platelet specific antibody, both the tests are negative. and less than 90% of the crossmatch are compatible, then there is no problem. Then if it is greater than 90% crossmatch are compatible, then you go for an ABO identifiable or incompatible or compatible platelets. Now, this is a basic thing which you can adopt as a diagnostic algorithm. But what we do as a process is based on the modalities that are available in your lab. And similarly, if we are suspecting a platelet refractoriness, what we do is upfront we do an HLA antibody screen, because most of the cases are of HLA antibody testing. So if it is positive, obviously we go in the direction of HLA. So basically we do a platelet crossmatch upfront and see if it is compatible or incompatible. If it is compatible, obviously, then we issue the unit. But however, if it is incompatible, we look for whether it's few units or if it is greater than 65% are incompatible. So if it is less than 35%, more often than not, we'll be able to provide the platelets to the patient. But if it is greater than 65, it is very difficult, and then you can resort to other modalities like HPA and ASP. So basically, you have to identify whether it is because of that or not. And many a times when there is no HLA antibodies, you go for platelet cross-match, and still, if it is incompatible, it may be a HPA antibody, and there also platelet cross-match is very handy. However, if it is compatible, you can issue the same. So this is a fine tweaking of the thing that we have done in our lab. So prevention of alloimmunization and thereby prevent platelet refractoriness. So what do we do to prevent the alloimmunization and thereby platelet refractoriness? You go for apheresis, leukoreduction. Now there are few modalities like ultraviolet radiation, pathogen reduction technology, and some recent advances in which they have tried to prevent the alloimmunization. So how do you prevent alloimmunization? The easiest way is if you have less of exposure, because single donor will cause, obviously, like we say, apheresis is going to be less of alloimmunization can be caused because of apheresis, because you are exposing less of donors. So this apheresis allows collection of platelet units that are from a single donor rather than a pool of whole blood derived platelet concentrate. So this limits the antigenic exposure. Similarly, leukoreduction. So basically, we all know the benefits of leukoreduction. One of the benefits is it prevents HLA alloimmunization, because most of the WBCs are removed by 3 to 4 log leukoreduction. And third modality is when you have a UV radiation. So ultraviolet radiation inactivates HLA class 1 and 2 bearing cells, and this causes decreased incidence of alloimmunization and thereby platelet refractoriness. This is not approved as of now. Pathogen reduction technology also is considered that it is helpful to reduce the immunogenicity of residual leukocyte, although that has also not been established that way that it can be used as a modality. There are other things that are recently, available. There is a lot of work going on to strip platelet concentrate of HLA by using the chloroquine diphosphate or a citrate buffer. And lower antigen expression may render cells in platelet concentrate less susceptible to recognition. Now, how do we prevent alloimmunization? These are the newer things that have come. One of them is human-induced pluripotent stem cells, which are a source for producing autologous HLA-matched or HLA universal donor platelets. This can be produced without donor dependency and possess the potential to eliminate alloimmunization to HLA and maintain shortage of platelet supply. So how do you overcome once the alloimmunization that is causing the platelet refractoriness? So there is a limited success in that, but there are few mechanisms by which you can do that, and there are like methods to neutralize the recipient allo antibody by adsorption or absorption, or a temporal blockade of immune mediated destruction. There is suppression of immune response or provision of modified platelet or alternate hemostatic compounds. That are there are lots of work going on, but there is no limited success in that, and so you have to there's a long way to go in this. So overcoming established alloimmunization, there are theory that alloantibodies can be absorbed by large platelet dose transfusion, repeated transfusion, and this can be done by in vivo adsorption, which will yield better increment count responses, as the corresponding alloantibody should no longer be present. You can even use a high dose IVIG to suppress the alloimmunization. RHIG infusions have been used to study to prevent refractory response. And there are other recent methods which are still under research, like use of vinblastine loaded platelet transfusion, cyclosporin A, immunoabsorption using staph A columns, plasmapheresis, bortezomib, eculizumab, which is a complement inhibitor, and acid treatment of platelets. The overcoming established alloimmunization with substitutes and alternative, you can have potential platelet substitute like lyophilized platelets, infusible platelet membrane, thromboerythrocytes, and thrombosphere. You can have growth factors such as interleukin 11 and recombinant human thrombopoietin. Basically, these are modalities that are still under research and they have not been established yet. And but for the sake of answer, you can always write these things and which are still under research mechanisms. And limited studies of newer thrombopoietin mimetic agents are also now available for refractory patients. And there is also an adjunct role of antifibrinolytic and recombinant coagulation factors. The facilities at our center presently, we do HLA typing, so we can provide HLA matched platelets. We can do HLA antibody screening and single antigen bead identification. So we have a virtual cross match literally. So ASP can be done, and we also have the facility of platelet cross match through the solid phase capture P method. We also have a modality known as platelet PAC-LX assay, which is a Luminex based assay to detect both HLA and HPA antibodies. And this is our experience. If you see, we have done this study, we can see that overall age group, if you see, the frequency is 34. We have had uncross match versus cross match 1 hour and 24. If you see, the p value is significant between the cross match platelets and uncross match platelets. This is the cross match platelets have a better yield and better CCI when we, especially in the patients who are platelet refractory. So these are the various conditions in which platelet refractoriness was noticed. It is a small study, but there is an ongoing thing going on for that. And this is the PAC Luminex based test that we perform in our center. And you can see that there is a negative control and it detects both HLA class 1 and the HPA antibodies. So here you can see the HLA class 1 is positive. And so what you're doing is you are also knowing the specificity, whether it's HLA or HPA in one go. And it is a handy thing, but requires, of course, it's an expensive test. So basically, if your platelet cross match is positive, then you can look for the specificity, whether it is HLA or HPA. So depending if you have the technology, you can always resort to PAC-LX, Luminex-based platelet and HLA antibody test. Thank you so much. I hope this was helpful. If there is anything that needs to be answered, you can always write in the chat box.